So good afternoon, everybody, and welcome to this, your third microbiology class. And today we're going to see, we've been talking about macromolecules, etc. Some of those macromolecules, by the way, we will return to, especially the nucleic acids. But today we're going to see how all of those macromolecules are put together into the form of the cell. And the cell is the basis of all living things on the planet. In prokaryotes, bacteria, no less than anything else. The basic unit of life is the cell, and all of its processes are directed by this hereditary material, and that is the DNA, which is contained somewhere in the cell. We're going to talk a lot today about the structure of bacterial cells, and then some as well about the structure of eukaryote cells. Even though we don't spend that much time studying eukaryotes in microbes in this class, we do refer to them every now and again. Nonetheless, it's very important for you to understand how how bacterial cells, for example, are related to the structure of higher cells, the eukaryote cells. So first of all, remember that bacteria fall into the group known as the prokaryotes. The cells, they share this characteristic with another group called the archaea that we don't talk about very much. Prokaryote cells lack a nucleus, and they have very little internal structure. They do not have membrane-bound internal parts, like organelles, etc. Those do not occur in the prokaryotes. No membrane-bound organelles. The most prominent organelle in the eukaryote cell, in the higher cells, is the nucleus. You will never see that in a prokaryote because they don't have organelles. They only have a cell membrane around. The only plasma membrane is the cell membrane around the outside of the cell, which describes the cell. There are some other differences as well. In the prokaryotes, the chromosome, the DNA hereditary material, is contained in a chromosome, but the chromosome is a circle. It is a very large circle. It's, if I was to take even a small cell like E. coli and unwind the circle of DNA, it would be more than 1 millimeter in diameter, that little circle. So that's an awful lot of DNA to get into a tiny, tiny, tiny cell that is 5 microns or less in length. The DNA is not contained in a membrane. It is inside the cytoplasm. I don't like to use the term that it is loose inside the cytoplasm. You can think of it like that if you want, but to say it's loose in the cytoplasm implies that it's somehow chaotic or it's just tossed in there. It's not. It's very carefully organized. And it has to be, because the cell has to be able to access the right regions of DNA. It has to be able to access the genes efficiently. And in order for that to happen, the DNA must be carefully organized, packaged in an organized way. As I've said, you've got to put an awful lot of DNA into that tiny cell. So it's very carefully wound up and packaged in order to fit in. All cells have a cytoplasm. So prokaryotes have a cytoplasm just as eukaryotes do. In addition, all cells have these little tiny, they're not organelles, but they're little tiny bodies called the ribosomes. So all cells have ribosomes, prokaryotes do as well. One of the characteristics of most bacteria, 99% of the bacteria, is that they have a cell wall. Now, this is a distinction which you must get clear in your mind. The cell wall is not the cell membrane. It's totally different to the cell membrane. It lies outside the cell membrane, and we'll discuss it in more detail in a minute, because it's a very important feature of prokaryote cells. The cell wall lies outside, and it contains, in prokaryotes, it contains a substance that is called peptidoglycan. It's a very, very strong substance. It's a polymer, and it's used to make a mesh-like cell wall that surrounds the cell on the outside of the cell membrane. So the cell membrane is on the inside of the cell wall. bacteria may have numerous other things associated with them. They may have flagellae, all sorts of extensions from the cell that penetrate through the cell wall and out, all sorts of things, excuse me, that we'll talk about in a minute. Excuse me. So the bacteria that we are going to talk about, the eubacteria, which are the true bacteria, those that distinguishes them from the archaea. Excuse. They include things like the true bacteria and the cyanobacteria and things like that we'll hear. There are many, many groups, but they all share this particular these particular features. Okay, so let's have a look at the actual structure of the cell. And in these diagrams here, this is just a three-dimensional diagram of a cell. Here is an electron micrograph, a very thin section stained of a bacterial cell. And this is a typical form. This is a form of cell which we would call the rod. So here is the cell membrane here. It's a plasma membrane. It's the only one, and it forms a cell membrane around the outside. You can refer to it as the plasma membrane or the cell membrane. I don't mind. On the inside of that membrane, remember the responsibility of the membrane is to allow conditions to define the cell and allow conditions inside to be different to the outside. So on the inside, there is a solution of biomolecules and salts and all sorts of other things. Very, very dense, very densely packed with molecules, so it forms almost like a gel, and that is the cytoplasm. And suspended in the cytoplasm, you can see these little bodies here. These are all of the ribosomes. And the ribosomes are responsible for producing protein. They're the ones that actually translate the message from the DNA. They are the ones that ultimately produce the protein, and we will eventually see exactly how they do that. Now, the DNA, as I told you, is inside the cytoplasm. It's very, very carefully organized. It's carefully packaged, and it will often form a region which may actually in some bacteria be visible even under the light microscope. Here it's very easily visible under the electron microscope here. There it is here. And this is all of the DNA. And it's carefully packed and wrapped and packed into this region called the nucleoid. Not a nucleus because it has no membrane. Instead, it's called the nucleoid in the prokaryotes. So remember, here's the plasma membrane. Right on the outside of the plasma membrane, you can see here in deep brown, that is the cell wall. And the cell wall has some particular responsibilities. It definitely does provide some protection to the cell. It provides some shaping to the cell, but its main responsibility actually is for osmotic control. And I'll explain that in a minute. So the main responsibility of the cell wall is for osmotic control. And there's the cell wall. Now, on the outside of the cell wall, there may be all sorts of other things as well. Very, very often, the bacteria will excrete a layer of mixed polysaccharides and substances that form a distinct non-living layer around the outside, which is variously called the capsule or the slime layer. A capsule is denser and sits more tightly against the cell. A slime layer is much the same collection of substances, but it is loose and easily removed from the cell, and literally does form slime. Sometimes if you grow these bacteria like that in culture, you'll see that the culture will actually become slimy as this layer of polysaccharides is lost from the cells. And there are several different functions for capsules and slime layers. And the two which are really important in health sciences. The first is very often capsules are for attach the bacterium to a surface, and also they often provide protection from immune attack. So it's a way of shielding, of the bacterial cell shielding itself from immune attack. We'll see that in pathogens, the capsule can often be very, very important because the first stage of infection is usually attachment on a host, attachment onto a host cell surface. There are various other things that can occur inside the cytoplasm. We may see different inclusions, for example, and very often these inclusions are storage products. For instance, they may store polysaccharides, they may store things like glycogen, and these may form distinct granules inside the cytoplasm. They are there to store fuel and molecules that the bacteria may need. In addition, the DNA in many bacteria in the nucleoid is not the only DNA present in the cell. Often what we will also find in the cell are the small circles, small, like little chromosomes, small circles of DNA, which are called the plasmids. Plasmids often contain very, very useful information for the cell. They may contain information, for example, which confers resistance to an antibiotic, and things like that. They may even contain recipes for capsules. In other words, they can often assist a bacterium. And the important thing about plasmids is that they are transmissible. When this cell dies and bursts in the environment, it may release its plasmids, and those plasmids can be taken up by other bacteria. They can acquire information from these plasmids. And you'll realize if this is carrying a gene for resistance to an antibiotic, this is one way that we can spread that information through a population of bacteria. Plasmids are something that are extremely interesting, and we will learn a bit more about them later on. The other thing to note about the bacterial cell is there may be extensions from the actual cell itself, from the cell membrane, in fact, out into the environment, which extend through the cell wall and through the capsule even and out. All of these that look like hairs are called fimbriae. They are proteinaceous, and they often are used also for attachment. And there are various functions for them, but their principal function is for attachment. We may also see actual structures that look like tubes called a pilus, which extend out. These also can have various functions, but the most important one is that these can be used to latch onto another bacterial cell and allow DNA to pass from one cell to another. And when that happens, that is a process called conjugation. And the pilus used then is called a conjugation pilus. It's a tube made of proteins. Very, very frequently, bacteria are motile. And they are motile by having attached to the cell these flagella. The flagella can be placed in various places around the cell. And you'll see it looks here like they are sinusoidal, like that. In actual fact, they are corkscrew shaped. It's a lazy corkscrew. And they are what is responsible for conferring motility on the cell. Very complex things. And we will talk a little bit about them in a while. But here you can see a clump attached to the end. So these are polar flagellae attached at one end, but they can have different bacteria will have different dispositions of flagella. They may be all over. They may extend all over. They may only be one. There may be a clump like this. It depends. It is characteristic for the bacteria as to what kinds of flagella they have. Let me just check. I think that that's about all I need to tell you for the moment. Okay, so let's have a look now at the different kinds of cells that you will see, different kinds of bacterial cells that occur. These are very important characteristics for classifying bacteria, and that is their cell form and their cell arrangement. The two are not the same thing. The cell form, first of all, this is a rod, and a rod the rod has one dimension which is longer than the narrowest. What I mean by that is, have a look at this distance here. The whole cell is much longer than that distance across. Rods can be very distinct like this, no mistaking them, but sometimes you'll see bacteria which are only slightly longer than they are wide. They're still a rod. Sometimes you'll see people refer to them as being a coccoid rod, which means you can't quite distinguish as to what they are. But this is a rod, and the proper term for a rod is a bacillus. It's a little bit confusing because there's actually a genus of bacteria called bacillus, which are bacilli, of course. But all rods are bacilli. So the coccus is a spherical or nearly spherical cell. And in this case here, you'll see that these cocci are arranged in a long chain. And those are streptococci. Strepto, straight, cocci, spherical cells. The streptococci are cocci which are in a straight chain. These are staphylococci. They're also coccoid, but they occur in bunches. Staphylopart actually is Latin for grape. They are like bunches of grapes. That's the easiest way to remember. This here is a spirillum. These are often large cells, and they have this distinctive sinusoidal shape like this. Very often they have a clump of flagella, very, very big flagella at the end here. And spirilla, most often free living. There are few pathogenic ones, but very common in marine systems and in freshwater, in aquatic systems. This here is a curved rod. It's still a rod, but it's curved into this almost like a C shape or comma shape. That is its proper term, is a comma shape. And these are often referred to as being vibrioid. And the reason that they're named vibrioid cells is because there is a genus of bacterium, Vibrio, very important genus. It's the genus there's a species of Vibrio that causes cholera. And those cells are typically curved like this. Although sometimes it's not that easy to see. But nonetheless, these are curved rods or vibrioid cells. And the last one is this. It's called a spirochete. And you may think it looks like this, but it's actually quite different. And what I want you to do is I want you to have a look really carefully in the center here. Can you see that it looks almost like a rod running through the center there? That is a bunch of modified flagella, which the cell is wound around, and it's wound around in a tight corkscrew all the way down. These can be very long cells. And this is a spirochete. And the spirochetes are very common. There are some very important disease-causing organisms that are spirochetes. One of them is Treponema pallidum. Treponema pallidum is the causative agent of syphilis, and it is a spirochete. But there are many spirochetes which do not cause disease. For instance, they're very common in our mouths, around our teeth. They're part of the normal microflora of our teeth. They have a very specialized form of movement. Many rods, the spirilla and curved rods, are actually motile. They have flagella, but the flagella point to the outside. And one of their characteristics is they can only go in one direction. They either travel in one direction or they kind of spin around. We'll discuss how they move in a while. But the spirochetes are different. The spirochetes, they have adapted their flagellum and it runs, that's what you're seeing, that rod-like thing running down the middle. They've adapted that flagellum and it enables them to spin in the medium. And they can actually move, and they can move forward, and they can move backwards. They're the only bacteria that can do that. They can move forward and backwards. They have a very distinctive type of movement. Cocci are never motile. They are never motile. And so if you see something that looks coccoid and it's motile, it's a coccoid rod. It's not a true coccus. Okay, so let's have a look here. We have referred to this before when we talked about biomolecules, but I'm going to run over again the structure of the cell membrane. And there's the plasma membrane and cell membrane. This is the only membrane that the bacterial cell has. You may occasionally see a bacterial cell which has extensions of the cell membrane into the cytoplasm. That is, you may see that, for instance, in photosynthetic bacteria, but they are extensions of the cell membrane, they're not organelles. So let's have a look at the structure again of the cell membrane. And remember that it is made up of these molecules, which are a specialized lipid molecule called a phospholipid. The plasma membrane is a double bilayer of phospholipids. And the phospholipid consists of fatty acid tails, here they are here, which are highly hydrophobic. And then it has a head. And the head is composed of glycerol. And it also has attached to it a phosphate group and another amine group, which we don't need to worry about. All I need you to remember is that this fatty acid chain here is highly hydrophobic, but the head here, especially the glycerol and the phosphate, are very, very hydrophilic. They dissolve in water very, very easily. So this creates a schizophrenic kind of molecule. One part of it wants to dissolve in water, the other part wants to escape from water. And if we put it into water, the only solution for the molecule is to form this kind of structure. And that is a bilayer of some sort in which the heads, here the hydrophilic heads, are dissolved. Remember, there's water here and there's water here. So the heads are dissolved into water on either side. And in between here, the tails extend in and they exclude all the water. So the tails are happy, no water. Heads are happy, dissolved into water. And what we see is this bilayer, which is absolutely typical of the plasma membrane, whether we're talking about the cell membrane or in eukaryotes, the membrane around organelles, we see the same structure. We see this one layer here and another layer here, and in between there, that is where the tails of the molecules are. So this, get it clear in your head as to what the driving forces are that lead to the formation of this kind of membrane. That on its own, I must add, is not a living membrane. The membrane on its own, this phospholipid membrane bilayer, is a very, very good barrier. There are not a lot of substances which can make their way across that membrane. There are some very small molecules can, and water can, but there are few substances which easily move across that barrier. And so what happens is that the cell places into that membrane many different proteins with different responsibilities, but their primary responsibility is to govern what goes in and what goes out. So they control the composition of the cytoplasm relative to the composition of the surrounding medium. This is, by the way, is not a solid, it's a fluid. These molecules move, they move the whole time. It's actually a fluid, and this is called a fluid mosaic model. The fluid mosaic model is this bilayer with its embedded proteins. Now, the chief feature of this membrane is that it's selectively permeable. That is, it only allows certain things to pass across very easily. And it will allow certain chemicals to enter and leave the cell. But most of the passage of molecules over back and forth is governed by the proteins which are embedded in it. So these are the membrane proteins. And some of them go right the way across there, like that. And those are called the integral proteins. Sometimes there are proteins which are only partially embedded, and they can be partially embedded on either side. But those are called peripheral proteins. And the ones we're particularly interested in are the integral proteins. And these proteins perform many functions. For example, I told you there's a cell wall on the outside. Well, that cell wall is actually secreted by proteins. It's created by proteins embedded in the membrane. In prokaryotes, all of the respiratory functions and photosynthetic functions, all of these sort of things take place at the cell membrane. And all of those proteins, very complex protein systems, which are associated with things like respiration and photosynthesis, those are embedded in that plasma membrane around the outside. In addition, what you need to realize is that a cell, a prokaryote cell, or any kind of cell really, has no eyes and it has no ears. It is alone, sitting in a medium, but it is essential that a cell be cognizant of what is happening in the medium around it. What is the pH? What are the concentrations of different ions? What is the concentration of nutrients? What is the concentration of sugar, et cetera, et cetera, et cetera. It needs to have information from the medium in which it is suspended. And that information is conferred by proteins. They are actually able to sense what is happening around them. They are also able to sense stimuli of other sorts, but they very often are measuring what is going on in the environment, and they will convey that information to the cytoplasm. They're going to convey that information to the rest of the cell so there can be a cell response. And then lastly, the proteins are responsible for transport. They're responsible for governing what passes across the membrane in either direction. Sometimes we'll see that transport is simply passive. It does not involve any expenditure of energy, but nonetheless, the proteins will be like a guardhouse. They will allow only certain things to pass across passively into the cell. Other times, the proteins will actively transport stuff. They will actively grab stuff and move it in one direction or the other. Most often, proteins are grabbing rare substances in the environment and pushing them into the cell and concentrating them inside the cell. And that is a process which requires energy, so that is an active transport system. We'll talk about it in more detail in a while. Okay, we need to spend a little time talking about the cell wall, because the cell wall is a feature of most bacteria. There are a couple of bacteria that do not have a cell wall, but they are few and far between. Most bacteria have a cell wall, and it has various functions. First of all, I should explain to you that it forms, as I told you before, it forms like a mesh. And the mesh is very open. The mesh has no control over controlling substances passing back and forth. It's just like a net that all the solutes can pass across quite easily. So that's not its responsibility. Instead, the cell wall does confer shape to the bacterial cell. That is true. And the cell wall, by the way, is not static. It's continually broken down and rebuilt as the cell grows, and then as it divides, etc. The cell wall is also restructured all the time. But it does produce a rigid structure which provides shape, and certainly will provide some protection from injury and from damage, maybe from predation. But its major responsibility is to prevent the cell from bursting. So let me just go back here and tell you that water can pass across this membrane very easily. In fact, there are sets of proteins which are there specifically to allow water to pass back and forth across the cell. But the other thing you need to realize is that the cytoplasm is very, very concentrated compared to the outside usually. So the cytoplasm has a lot of stuff in it. It has a lot of salts, a lot of molecules, etc., etc., etc. In other words, if that is true, then it has much less water than the outside. And the laws of diffusion will tell you that diffusion tends to even out the concentration of substances. Now, the cell will not allow the solutes to leave, but it will allow water to pass in. So there is a tendency now for water to continually pass into the cell across the membrane. It is attempting to dilute the interior to get it to the same concentration as the outside. That form of diffusion, the diffusion of a water molecule, is called osmosis. So osmosis continually pumps water into the cell in an attempt to dilute the interior of the cell. This is a huge problem because that cell membrane is fluid, it's fragile. And if you allow water to pass into the cell continuously, then the cell begins to swell like a balloon. It will start to swell, and it will reach a point where the pressure is so great that the cell will burst. And this is a real and present danger for cells, for bacterial cells especially. Because eukaryotic cells often, they suffer the same problem, especially if they're in a dilute medium like fresh water or whatever. They suffer the same problem, but eukaryotic cells have specialized systems, specialized organelles and everything that pump water out as fast as it comes in. So they don't face the same problems often. The bacteria have no way of doing that. They have no way of pumping water out. The water continually makes its way into the cell and swells the cell. So what they have done is the cell wall has evolved as a protection, because what happens is that the cell begins to swell, and it will swell up against the cell wall, and the cell wall does not give way. The cell wall allows pressure to build up inside the cell without the cell bursting. And the way to think about it is this: that water, the diffusion of water into the cell, that is a pressure. It's pressure that is pushing that, and the pressure goes from high to low, right? And that is the osmotic pressure, from high outside, lots of water outside, and the pressure is to push water into the cell where there is only a little water. So that's the osmotic pressure. The cell wall allows pressure to build up in the cell until the pressure inside the cell equals the osmotic pressure. And when that happens, then as every time a water molecule goes into the cell, a water molecule goes out. It establishes an equilibrium. Effectively, you can just think about it in common sense terms. The cell has swelled up against the cell wall until it can go no further, but it's not going to burst because the cell wall keeps it in. But it does mean there is a pressure inside the cell, and that we call the turgor, T-U-R-G-O-R, the turgor pressure. When the turgor pressure equals the osmotic pressure, everything's fine, because as much water goes in will go out. That's the principal responsibility of this cell wall. It is an osmotic, it is a protection against osmotic stress. The cell wall molecules are important in disease processes. They do have antigenic properties. That is, they can be recognized by the immune system. And the cell wall can actually be attacked by various elements of the immune system. So they're targets of the immune response. And in modern times, many of our antibiotics, many antimicrobial strategies are directed against the cell wall and against cell wall synthesis. And a really classic example is the very first member of this whole group was penicillin. And what penicillin does is it interferes with peptidoglycan synthesis. So it ends up weakening the cell walls of cells, of bacterial cells. And when it does that, they burst from osmotic pressure and die. That is how the penicillins all work. But in natural systems, first of all, penicillin is produced by fungi for exactly that reason, to kill bacteria in the fungal environment. But our own bodies produce substances which will attack peptidoglycan in the cell wall. And the most easily understood is an enzyme called lysozyme, which occurs in tears and other body fluids as well, but its classic location is in tears. And it attacks the cell walls of bacteria, and thus is an antimicrobial. It's a disinfectant in our tears. And it's very important in keeping the eyes free of infection. Okay, now we're going to have to talk a little bit more about the actual structure of the cell wall. This peptidoglycan that is in the cell wall, it's a disaccharide, but it ends up forming these long fibrils and chains which get linked together into layers, and all of the layers can be connected to one another, producing this very strong mesh network. I don't need you to pay a lot of attention to the biochemistry of the peptidoglycan, but I just want you to understand that it's capable of forming polymer, which is extremely strong. But it is the way in which those polymers are put together that becomes very important and very distinctive. There are two distinct groups that we recognize. There are two distinct types of cell wall, and those are referred to as gram-positive and the gram-negative cell wall, and their structure is quite different. They have taken different evolutionary paths. As I told you before, there are a small number of bacteria which have no cell wall. which we'll refer to sometime because some of them are really important, but they're a minor group compared to those which have a cell wall. Those that have a cell wall, either gram positive or they are gram negative. And here, for example, is a gram positive. And here you can see, first of all, let's orient ourselves. Here's the cell membrane here. Here's the interior of the cell, the cytoplasm. The plasma membrane here. And then on the outside, the cell wall. And gram positive has a thick layer of peptidoglycan. And you can see these several layers of these fibrils. And they are all tied together with a substance called teichoic acid. So they have a thick, rigid outer cell wall, and that is all. Get this straight because you'll see the difference in a minute. In the gram positive, there is only the plasma membrane, forming a cell membrane here, and then the cell wall on the outside, and it is thick. In the gram negative bacterium, the structure is completely different, and it acquires all sorts of different functions. So first of all, orient yourself. Here's the cell membrane here. The plasma membrane forming the cell membrane. On the outside, it still has some peptidoglycan, but it only has a thin layer, and they are not, there's no joining into two layers above them. So it's just a thin layer, does the same thing. The peptidoglycan cell wall still offers osmotic protection to the cell. It's still required, but it is very thin. And then on the outside of the cell wall, there is a very complex second membrane. At first sight here, it looks as if it's just a repeat of the plasma membrane. It's not. It's quite different to the plasma membrane, and it is called the outer membrane. And it does have phospholipids in it, but it also has various other substances, and especially in this outer sheet here, it has these substances called lipopolysaccharides. So in other words, they're lipid tails, but they have polysaccharide heads. And they have another lipid associated with them called lipid A. Now, the reason I emphasize those is polysaccharide and lipid A and the lipopolysaccharide, etc., is because these substances are very strongly antigenic. They can excite a very powerful immune response. And in fact, that response can be so profound that it is actually dangerous. If we have an infection with gram-negative bacteria and we start to get this stuff floating around in the bloodstream, we can end up in a very bad situation, which is a clinical condition called sepsis. And that is an overwhelming immune response to the presence of these in the body fluids. So gram-negative bacteria have this very complex cell envelope. That is what this is all referred to as. This is the cell envelope. And it consists, first of all, of the cell membrane, the ordinary plasma membrane, and then this outer membrane. And the outer membrane, it looks something like this, but in fact, it has very different substances embedded in it. It still has various proteins embedded in it, just like the plasma membrane that forms the cell membrane here. It does have various proteins, and they have very complex characteristics. One of the characteristics of these proteins is that they are able to select very rare, useful molecules out here in the environment, and they're able to move them into this space here. This space is important. It's called the periplasm. And it's the space between the outer membrane and the plasma membrane. And it can act as a storage area. The periplasm and the outer membrane also confer different properties to the bacterium. They're able to detoxify, for example, harmful substances inside the periplasm. So that it begins to sound like that this gram-negative bacterium has taken a great evolutionary leap, which it has done. This outer structure confers all sorts of very useful properties to the cell. So here, just to emphasize, there is the structure of that lipopolysaccharide. Here is called the lipid A that's embedded in the lipid bilayer. There is a polysaccharide here, and then on the outside is the O. This is often called the O antigen, and it's also a polysaccharide. O antigen because this is one of the things that excites a very, very, very strong immune response. So it's LPS, lipopolysaccharide, a constituent of the outer membrane of this lipid A is also very dangerous in an infection. We refer to it as being an endotoxin. That is, it's part of the cell itself, but it has a toxic function because of the reactions that it excites during an infection. This varies a lot. The structures of these vary a lot, not only species to species of gram-negative, but within gram-negatives. And so you may find, for example, one strain of an organism like E. coli, which is completely harmless and its outer membrane has no pathogenic quality whatsoever. But then you find another strain. Here is one that we're going to talk about much later on, E. coli 0157H7, where this outer membrane causes tremendous toxicity and tremendous problems when a person is infected with that particular strain of E. coli. Let me just, I just want to go back here and tell you a couple of things. The gram-positive wall you'll find in rods and in cocci, and you very, very seldom see a coccus that is not gram-positive, but there are some important exceptions that I'll tell you about in a minute. So you see gram-positive in rods and in cocci. Many of the well-known bacteria, Bacillus itself is gram-positive. Various other things, Clostridium, all these rods, these are all gram-positive rods. Gram-negative with this complex outside. The best known example of a gram-negative, as I just told you, is E. coli. But there are many, many others, which Vibrio, all of these organisms, these are gram-negative. One of the things about gram-negative bacteria is this issue of being able to concentrate rare molecules. This means that often gram-negative bacteria have an ecological advantage. They can survive in environments where nutrient is very rare, where other bacteria can't, because they can concentrate the nutrient against a concentration gradient, and they can concentrate it here in the periplasm for use by the cell. So many, many, many of our really dangerous pathogens are gram-negative, but equally many are gram-positive. Gram-negative because they have this thin peptidoglycan wall. They tend to be less sensitive to antibiotics, which hit at cell wall synthesis. We often use other antibiotic systems for gram-negatives. So I'll also just tell you, just very briefly, why are they called gram positive and gram negative? They're called gram positive and gram negative is named after somebody called Gram, who developed a staining technique. And it's a very simple staining technique. I'm hoping that you do have an opportunity in lab to do it. But it is a classical clinical staining technique to distinguish between gram positive and gram negative bacteria. All that you do is you stain it with a substance called crystal violet. That stains both gram negative and gram positive. You then add a little bit of iodine. And what that does is it binds the crystal violet very tightly to the peptidoglycan. And you rinse the cell with a solvent which dissolves crystal violet. What happens is in this, you do it very briefly. You have to do it very carefully, very briefly. You rinse it with a solvent for crystal violet. And the solvent in the gram positive, because there's so much embedded into the cell wall, it's unable to wash it all out. In the gram positive, however, when you wash it with the solvent, first of all, it disrupts the outer membrane, and it washes all of the crystal violet out away from the peptidoglycan and washes out any that's inside the cell as well. So this cell, when it's tested with the solvent, it goes colorless. You have to then stain with another dye. You usually use a pink dye, just so that you can visualize the gram negatives if it's gram negative. Gram positive stays black. And that is the blue-black color of the stain is what tells you this is gram positive. In the gram negative, all the blue-black has been washed away, and all you're left with is the pink color, and that is the classic gram negative stain, a pink stain. And it allows us to immediately distinguish between these two great groups of bacteria, which diverged a very long time ago and formed these two very distinct evolutionary trends. Okay, this is the one group of bacteria without a cell wall that we must be aware of. And this is Mycobacterium. I'm sorry, I just made a stupid mistake. Forget what I said about not having a cell wall. This is Mycobacterium. And Mycobacterium has a specialized cell wall. I'm sorry, it's not that it doesn't have a cell wall. Mycobacterium contains a very strange lipid in its cell wall. It's called mycolic acid, and it's like a wax. And because the cell wall is embedded with this kind of waxy substance, it doesn't react to the reagents that we use in the gram stain. It's very difficult to get a gram reaction from Mycobacterium. And instead, we have to use a special staining technique. It's called the acid-fast stain. And in order to do so, it's quite a complex staining procedure. But what you end up with, when you look at it, you end up with the cells stain, if they are Mycobacterium, they will stain a bright red color, and that is diagnostic of Mycobacterium. Mycobacterium is a very interesting bacterium, and there are numerous disease-causing Mycobacteria. The most important one, Mycobacterium tuberculosis, the causative agent of tuberculosis. There is another one called Mycobacterium leprae, and that causes leprosy. They have a characteristic, they are usually slow-growing, but as you realize, they can cause very, very serious disease, and they are resistant to many, many antibiotics. First of all, because of the structure of their cell wall, but second of all, because especially Mycobacterium tuberculosis is able to mutate and has done several times, and becomes resistant to the antibiotics that we use against it. Here it is. Here you see that the cell shapes can be very, very distorted, strange sort of cell shapes. Okay, and I'll just mention that there is another group. I'll only give you the name. This is why I got confused when I was talking. There's another group called the Mycoplasmas. Mycoplasma, it's another group, has no cell wall, and it has to actually adopt very strange strategies in order to survive without a cell wall. It also causes disease. We have a disease-causing mycoplasma. Okay, so remember that we're looking at that cell membrane. What we saw was that the cell membrane, the plasma membrane, consists of phospholipid bilayer, and embedded in it the proteins. And those proteins are what confer the living nature to that fluid mosaic that composes the plasma membrane. So the regulation of what passes back and forth is the responsibility of the proteins. And the first kind of control that we're going to see is although these proteins, which allow essentially free passage of molecules without expending any energy. These processes of transport are always a simple diffusive, a diffusive process, either simple diffusion, or the protein may make it a bit easier to occur, as I'll explain in a second. But they are diffusive processes. And so these passive transport proteins, they always allow transport only from an area of high concentration to an area of low concentration. So they will only transport stuff in the direction of the concentration gradient, from high to low. So the processes of simple diffusion, they will take place just across the plasma membrane. So, for example, there are some ions and things which can pass just across the plasma membrane. In fact, water can drift across, but it doesn't go across all that easily. But there are various things which can make their way across the bilayer. But for the most part, most things actually require a passageway to get across the plasma membrane. And this is a responsibility of those proteins which allow the facilitated diffusion. So these proteins, they're embedded into, they're integral proteins embedded into the membrane. They have a passageway through the middle of them, but they are specialized. They don't just allow anything to diffuse across. They are actually selective. So they are specialized to only allow certain stuff to get across. Some of them, it's a simple passageway all the way through. But in others, there is actually an active, not an active process. Active has a specific meaning that I'll talk about in a second. But they will actually grab onto the substance and push it across. And this is called facilitated diffusion. So these are facilitated diffusive processes. The protein is actually selecting particular solutes to go across the membrane. There's one class which are highly selective because they only allow water to go across. So these facilitate osmosis. And these are proteins, these are protein channels for water called the aquaporins. And there are many of them. It's one of the reasons why water passes across the membrane so easily. It can get across without the aquaporins, but it's impeded. It's a slow process because it has to make its way through this lipid, which is hydrophobic. Instead, with the aquaporins, water can move across very, very easily. I just want to mention something. Don't forget that this is a diffusive process. So it only allows from high concentration to low concentration. Should the concentration of a substance build up so high in here that it exceeded the outside, the substance would go the other way. Okay, but most of the facilitated diffusion will take this place in this direction, because most of those substances are probably rare inside the cytoplasm. So the other kind of transport is active transport, and these are proteins which actually require energy in order to move molecules. They require energy usually in the form of ATP. ATP we have not talked about yet, but ATP is the only molecule which, in this case, which will convert them useful biological energy. They break down ATP. If you remember back when we talked about the functional groups, ATP has a phosphate group on it. Lodged in that phosphate bond is a lot of energy. The enzyme, this protein embedded in the membrane, is actually an enzyme, and it is able to break down the ATP. And when it breaks down the ATP, it can grab onto rare molecules in the environment and force them across the membrane and push them into a high concentration area. So in other words, active transport is usually used to move molecules against a concentration gradient. The passive went from high to low. Those are diffusive processes. Active transport takes from low concentration to a high concentration because it wants to accumulate useful molecules inside the cytoplasm. But in order to move against that concentration gradient like that, it has to put in energy. So that is an active transport process. Okay, so that's, for the moment, the functions of the membrane. Let's have a look inside the bacterial cell. As I told you, the DNA is packed into an area called the nucleoid. And despite the fact that this is very large, this is actually a circle of DNA. It's the bacterial genome. It's all of the DNA that confers hereditary information consists of this giant circular chromosome, and then smaller genetic elements called the plasmids. The plasmids may carry very, very useful information, but they do not carry information which is essential to the normal functioning of the cell. It is additional information, additional useful information, but they are not crucial genes. All of those crucial genes, all the householding genes and everything else, they are on the bacterial chromosome. The plasmids can bring in all sorts of information. They can bring in antibiotic resistance. They can bring in information which allows the bacterial cell to produce toxins. They can bring in all sorts of things which allow the bacterial cell to acquire pathogenesis and become pathogenic. And so the acquisition of a plasmid can actually change the nature of a bacterium radically. We will learn later on that we actually we can manipulate this in genetic engineering. We can take plasmids and we can deliberately put them into bacteria carrying useful genes. Also in the cytoplasm, we will find these little tiny bodies called the ribosomes. Now, these are the sites of protein synthesis. They are remarkable little bodies. They are made up of protein, but they're also made up of RNA. And this is an example, I think I've mentioned before, that proteins, for example, forms most of our enzymes. 99% of our enzymes in the cell are protein, but there is an exception to that, and that is RNA. RNA can sometimes act as an enzyme. And when it does so, we refer to it as being a ribozyme. And the ribosomal RNAs are one of these, because it is the ribosomal RNAs which are responsible for driving the process of translation, which creates proteins reads off the message from messenger RNA and will create a protein. So the ribosome consists of proteins, and it consists of ribosomal RNA. It has specialized RNA, some of it enzymatic, the ribosomal RNA. And there are two subunits. There's a small one, and there's a large one. And normally inside the cytoplasm, they exist apart from one another. But as soon as messenger RNA appears, the ribosomes will latch onto the messenger RNA, the small particle first, and then the large particle, and they will begin to read the messenger RNA and produce a protein strand. Then we have various different inclusions inside the cytoplasm as well. Sometimes these are visible quite easily under the microscope. Other times they require special staining in order to show them. But the things can appear as granules. These are often storage things. It may be polysaccharide. It can be, for example, glycogen. It can also be phosphate, these sort of things. You sometimes see lipid droplets as well. In some specialized bacteria, they accumulate sulfur inside as bright yellow crystals. In cyanobacteria, which are photosynthetic, there are specialized inclusions, very large and easily visible, called the carboxysomes, which are involved in the process of carbon fixation. Then we got some others which are functional. They give the cell some environmental specialization. For example, aquatic bacteria sometimes have gas vacuoles, and those can be regulated so that this bacterial cell can regulate its position in the water column, up and down. Here's a really interesting one. This here are called magnetosomes, and they are iron oxide, and they're little tiny magnets. Now they do have some cellular functions, in particular that they're involved with the destruction of peroxides inside the cell. We'll hear about later. Peroxides are very dangerous in the cell, and their cells often have several mechanisms to break down peroxides. But the magnetosomes are extremely interesting. They're little tiny magnets, and they occur very often. These are bacteria which are in the substrates. So try and think of a function for these magnetosomes. Why would a bacterium have a little magnet inside it? Well, the answer is that if the substrate is disturbed and the substrate bacteria are whipped up into a water column, they have to find their way back down to the substrate. And what they do is they actually orient themselves with the Earth's magnetic field. The Earth, penetrating the Earth all over the surface, are lines of magnetism going to the core. And the bacteria actually orient themselves with a line, of a magnetic line, and move down it and find the substrate again. Just as we use a compass, a magnet in a compass, so the bacteria are able to have their own little compasses embedded in them to allow them to. Isn't that marvelous? I think it's fantastic. Okay, so what I'm going to tell you about now applies only to the gram-positive bacteria and only to selective gram-positive bacteria. There are certain gram-positive bacteria, particularly the genera Bacillus and Clostridium, which are able to produce a highly specialized cell during cell division. Normally, bacterial cells divide in response to rich environment. Everything they grow, they divide in the middle, and they produce two cells which are identical to one another, which then grow, divide again, etc., etc., etc. But as conditions begin to get, as the nutrient gets used up and as waste matter begins to accumulate, etc., the growth of bacteria begins to slow down. And in those conditions, these two genera, Bacillus and Clostridium, are able to produce a highly specialized cell. And it is called a spore. And it's produced by the formation, by a process called sporulation, in which the cell division is not equal. So here you see, this is a mother cell here. And the mother cell divides, but it divides unequally. You can see there's a small portion rather than two equal size cells. There's a small cell up here. That small cell becomes invested with a coat. I won't discuss the coat in great detail at the moment, but I'll just tell you that this coat is thick and it is highly specialized. There are two major layers to it. The spore forms inside the cell, so it's referred to as an endospore. We see other kinds of spores in other microorganisms like fungi. They produce spores outside, so they're exospores. The endospore is produced inside the bacterial cell. Uneven division of the cell. It has its own cell membrane around it, and the cytoplasm of the mother cell invests it with a thick spore coat. And that spore coat is extremely, extremely interesting because it confers all sorts of protections on the spore. This genus here, Clostridium, is very, very sensitive to oxygen. It cannot tolerate oxygen in its environment. It is completely anaerobic. But the spore can exist in oxygen. So the spore can be used to disperse the cells through the air until it finds an anaerobic environment in which it can grow. So first of all, the spore has just about no metabolism at all. It's metabolically virtually inert. The spores are extremely long-lived. We have recovered spores from graves that are thousands and thousands of years old and been able to culture the bacteria from them. The spore, in addition, inside here, there are sets of proteins which protect not only the DNA, but also the useful proteins of metabolism and everything. And the spore is very, very resistant to heat. Very often you can boil spores without damaging them. If we want to kill spores, we actually have to put them into a pressurized, heated environment, which in microbiology, in the lab, we use an autoclave, which is basically a pressure cooker. To raise it, when you pressurize them, you can raise the temperature well above the boiling point, and that will kill them. That's the only way you can kill them. The spores are also very, very resistant to disinfectants. They don't react to things like phenol or any of these commonplace disinfectants that are used in hospitals. This means spores are extremely dangerous in health terms because the spores can actually last a long time. They can be dispersed easily, and they're very often disease-causing organisms, both Bacillus and Clostridium. So spores were the major source, a major source of contamination in hospitals and things like that, in surgery and everything. Even if they boiled instruments and everything, it didn't kill spores. It killed living cells, but it didn't kill spores. And so it's very difficult to sterilize completely. Instead, we have to sterilize in an autoclave to get rid of the spores. Other features of the outside of the cell. First of all, I told you right at the beginning, sometimes there is a looser layer of polysaccharides outside the cell. In this case here, they are sitting closely adherent to the cell, and this is a capsule. You use a special staining technique to visualize them. Very important feature because often it confers pathogenicity. on a cell. It enables the cell to stick to a host cell. Capsules are well organized and they're quite distinct, but sometimes it's a loose layer, and that we call a slime layer. It's often referred to as a glycocalyx. That simply refers to the composition of the biomolecules that make it up. These are often glycosylated. They're polysaccharide, have lots of polysaccharide groups on them. It's a polysaccharide, largely a polysaccharide. May have some protein with it, but it's largely polysaccharide. So here are the flagella, which confer motility on cells. There are no motile cocci, only rods of one sort or another, or spirilla. And here are the flagella. And what we can see here, for example, this cell here has flagella coming out all over the cell. Those are referred to as being peritrichous. This cell here is monotrichous. It only has one, and that is polar. Is at the end here, not peritrichous. Lophotrochus is a bunch of flagella. Now, Lophotrochus and polar. And these are kind of specialized. They're called amphitrichous flagella. They're actually very, very large flagella in a bunch. And that's characteristic of the spirilla. They're amphitrichous and they are polar. Now, the flagellum structure, I won't do it in any great detail, but it's just really interesting to look at. It might be tempting when you look at this flagellum to think that it's beating like this. That it's a sine wave, right, moving down the flagellum, which is propelling the bacterium forward. It's not. In actual fact, the flagellum is shaped like a corkscrew in space. And at the bottom, it has this ingenious little motor, which is embedded in the plasma membrane. In gram-negatives, it's embedded in both the plasma and the outer membrane. But here's a gram-positive. So it's just embedded in the plasma membrane. And that little motor actually spins it like that. So it's like a propeller spinning. And that's how it propels the bacterium through the water. We'll talk later about how this actually works. In addition, on the outside, we may have these long extensions of the cell membrane called the fimbriae and the pili. And fimbriae are these thread-like proteinaceous extensions, and they usually are to attach to the substrate. That's the usual function. And they may be very important in establishing infection because the cell has to first sit on the host cell. It has to attach firmly to the host cell. Pili are much longer. They have various functions. They're usually tube-like protein extensions. And one of the interesting things about them is they confer another kind of movement on cells. And that is sometimes cells will be seen to twitch across a surface like this. And that is through the action of pili. Also, some bacteria, some organisms move in a slow gliding fashion that has nothing to do with flagella. Instead, it's also due to the pili. They can confer motion on a cell. But remember, one of their really important functions is to transfer DNA, hereditary material, between cells in the process of conjugation. Those are sex pili. Okay, I'm going to stop there, and I'll add this little section on eukaryote cells to your following class. Okay, but you can stop here.